I'm very happy to present the first speaker of this afternoon session, Marco Demaria. Please welcome to the stage and go ahead whenever you're ready.
All right, so good afternoon, everybody. So first of all, let me thank the organizer for putting together this fantastic meeting and also for giving me the opportunity to share with you some of the recent data from the lab. And as a personal announcement, I heard that there are millions of dollars flying around for aging research. So our lab is very happy to accept any money that you want to give us. So please talk to me if you have any. So we have heard already quite a bit about senescence during this meeting. So I don't have to go too much into the detail during the introduction, but just as a reminder, senescence is a state that is essentially a state of stable and generally irreversible growth arrest, which depends on two potent oncosuppressor proteins, which are p53, and the cyclin-dependent kinase inhibitor p16. So senescent cells undergo a number of quite heterogeneous morphological, structural, and functional changes, including the activation of this secretory phenotype, which we call the SASP, which has quite a prominent pro-inflammatory signature. So we have also heard in different ways that senescent cells accumulate with aging. Here is a very simple example of this. This is a mouse model called P16-3MR. This is a mouse that we have generated when I was in Julie Campisi lab, so almost a decade ago now. Where you can clearly see that with increase of time, you start to see more and more of this luminescent signal that reflects the abundance of P16 positive cells. So the more you age and the more P16 cells accumulate in your tissues. But also we have learned, also during this meeting, that senescent cells are associated with virtually any chronic dysfunction that we see in aging. So you can go from neurodegenerative dysfunction to fibrotic disease to cancer, you name it. Senescent cells seem to play a role either for the onset or for the progression of the disease. And this, of course, sparked this interest in developing xenotherapeutics, so interventions that could interfere with the detrimental function of senescent cells. However, there is senescence is a quite complicated phenotype. It's not at all an homogeneous phenotype. So there are different subtypes of senescent cells that express different type of features. And a few years ago, Alejandra, a PhD student in the lab, decided to analyze a number of transcriptomic data to try to see how much indeed heterogeneity exists between different subsets of senescent cells. And to make a long story short, indeed, there is an enormous heterogeneity. So every subtype of senescent cell expresses a very different signature in terms of transcriptome. However, there is also what we call the core signature of senescence or common signature of senescence, which are these 55 genes that we see somehow equally distributed among downregulated and upregulated genes that seem to be indeed represented in all the subtypes that we have looked at. And when you try to categorize the function of these genes, you can see categories that totally make sense with the senescence phenotype. So you see downregulation of genes that are part of the cell cycle progression, which reflect the state of cell cycle arrest, which senescent cells are. And you see in the upregulated category genes that encode for proteins related to inflammation, which of course reflect the SASP and this pro-inflammatory phenotype. Genes that are related to the p53 signaling. And also, interestingly enough, genes that are involved in apoptotic modulation. And in particular, you see that there is the upregulation of genes that are both pro and anti-apoptotic. So essentially, the senescent cells seem to be primed to apoptose, but at the same time, they mount this brake to the apoptotic cascade because they upregulate these anti-apoptotic genes. And among all of them, we actually see one that is always up in senescent cells, which is BCLW, which is encoded by this gene that is called BCL2L2. So we reason that perhaps if you try to remove this anti-apoptotic machinery, then the senescent cell would spontaneously enter into apoptosis, right? And we did that in collaboration with Dao Hongzhou, which at the time was at the University of Arkansas, now it's at the University of Florida, where we took different models of senescence, and we treated these cultures with ABT263, and what we could see is that indeed these cells started to lose viability, suggesting that we were killing the senescent cells. And importantly, this compound, which is called ABT263 indeed, was non-toxic for non-senescent cells. And ABT263 targets and inhibits 3 different proteins, BCL2, BCLXL, and BCLW, and they are all 3 proteins part of this anti-apoptotic machinery. Importantly, we also demonstrated that this compound can eliminate senescent cells in vivo. So now you take our reporter mouse, where you see this luminescent signal that comes up when, for example, you expose the mice to high genotoxic stress, in this case, when you inject the mice with this chemotherapeutic agent called doxorubicin. But then if you do the same and after that you treat the animal with ABT263, you see that there is this removal of luminescent cells, so suggesting that you're killing selectively P16 positive cells. And it's important to say that the mice handle the drug very well, so there is no obvious long-term toxicities of exposing the mice to ABT263. So now we decided to use another model that to test the validity of our approach, and this is a model of skin aging, and actually a particular phenotype of skin aging. So you're probably all familiar with skin aging because it's probably one of the most common and well-described and better described aging phenotypes. And it's interesting because it's a phenomenon that occurs because of both intrinsic factors, so just the pass of time, so chronological aging, but also because of the exposure to extrinsic factors such as UV radiation. And there are several features that are indeed that have been very well described that happen with skin aging. You see that there is a loss of epidermal thickness, a loss of cellularity, there is loss of subcutaneous fat, there is wrinkling because you start to lose collagen. And there is another interesting feature, which is that in general, aged skin become hypopigmented, so you lose pigmentation. But you increase the number of these age spots, which are essentially hyperpigmented lesions. There are clusters of these cells that secrete they secrete pigments, that is, in this case, is melanin. And these are normally melanocytes, and in particular, these are melanocytes that express senescence-associated markers. So now if you take a mole, which is one of the classical hyperpigmented lesions that we probably all of us have some on the skin, and you analyzed and you zoom in these structures, you clearly see that you identify these cells as senescent because you start to see a lot of senescence markers that come up in these cells. And these are essentially structures that are populated by senescent melanocytes. So our idea was, well, we have this drug that can kill senescent cells. And perhaps we can develop this drug in a way that we can use topically, and we can try to remove these structures. And it's important to note that, of course, cosmetically this might have a sense, also because these structures cannot be removed in other ways than not with surgery, so that's not something you would want. But also because, for example, from moles, with time, you increase your risk also to have cells that might escape and become melanoma. So it is possible, and it's predicted that 20 to 30% of melanomas arise from these existing hyperpigmented lesions. So we thought, okay, we can try to use this compound, this ABT263, topically on the skin and see what happens. And this is work that was pioneered by Jas, a postdoc in the lab. So Jas initially did a very simple experiment just to check whether the anti-apoptotic machinery was upregulated in senescent melanocytes. So he took a simple culture of senescent melanocytes and he measured the expression level of different members of the BCL2 family protein. And as in accordance to our transcriptomic data, he found that the protein that was the most overexpressed was BCLW, suggesting that perhaps these cells have mounted a sort of anti-apoptotic machinery, so they were resistant to apoptosis. And interestingly enough, we also look into human nevi to see whether they were also expressing this protein, and it was the same. So if you look in the nevi, you could see this upregulation of BCLW, so this anti-apoptotic protein. So we were very hopeful that the cells were sensitive to ABT263, but unfortunately this was not the case. So when we expose either non-senescent or senescent cultures of melanocytes to ABT263, we saw a very poor selectivity against senescent cells. So the senescent melanocytes were mostly resistant to ABT263. And we tried this with different types of senescent melanocytes. So we tried to make these melanocyte senescence by replicative exhaustion, by oncogenic stress, by genotoxic stress, you name it. All these type of models, they were resistant to ABT263, and they were resistant to an analog of ABT263, which is called ABT737. So then we decided to look a little bit more in what could protect these cells from dying, because they seemed actually also primed to apoptose. So by the transcriptomic profiling of these cells, they seemed they were also primed to apoptose, but still they were resistant to anti-apoptotic inhibitors. So to make a long story short, what we found was that essentially when you expose your senescent melanocytes to ABT263, and only your senescent melanocytes to ABT263, you start to see upregulation of another anti-apoptotic protein, which is called MCL1. And MCL1 works on a different pro-apoptotic cascade, which is activated by this apoptotic factor called NOXA. And we also demonstrated that this was due to enhanced translation. So we could see that there was a higher amount of MCL1 RNA carried by heavy polysomes. And also that this was dependent on cap-dependent translation, because if we now expose our senescent melanocytes to the treatment with ABT263 and to an inhibitor of cap-dependent translation, we could completely block the upregulation of MCL1. But then, of course, the real question is, does combining MCL1 inhibition to ABT263 now sensitize senescent melanocytes to die? And that's what we tried. So initially we tried with a genetic approach, so we used short terpene RNAs, and we knocked down the MCL1 protein, and what we could see was that now we started to have cells that were very sensitive to ABT263. So there was and this was really synergistic, because either of the intervention per se did not do anything to the senescent melanocytes. So there was synergy between these two approaches. And then at the time we started this, there were not real good selective and specific inhibitor for MCL1, but we were lucky enough that a new compound was developed, and this is called S63846. Historically, MCL1 has been a target for cancer treatment, but very difficult to inhibit. But indeed, this was published in late 2017. This compound. So we treated also the cells with a combination of this compound, so pharmacological MCL1 inhibition and ABT263. And now again, we could see that there was a very nice selectivity against senescent melanocytes. So then we decided to move to some more relevant systems and models. So the first one that we used is organotypic cultures of human skin. And this was done in collaboration with an expert on these cultures, which is Todd Ritke, which is an University of Pennsylvania. So Todd developed these cultures for us, where you clearly see this very nice 3D skin that has a dermal layer, an epidermal layer. And in these cultures, it's spiked in essentially senescent melanocytes that you can see here that are stained in purple. And then we exposed these cultures to our combination treatment. And what we could see was that these structures completely disappear. So these senescent melanocytes were killed from these cultures, but the skin structure were completely unaffected. So there was no toxicity for the other cells that were present in the tissue. And then at last, we did use even more physiological system. So we actually took a mouse. And this is a mouse where we can inducibly generate these hyperpigmented lesions that you can see here. You can start to see after induction these very nice black structures that are indeed this nest of senescent melanocytes that secrete a lot of melanin. And what we did is that we treated these animals topically with the combination of ABT263 and S6. And we could see that also in this case, we were able to eliminate these structures from the skin. And this was, again, very synergistic because we could not see such an effect with either of the drugs individually. So with this, I conclude, and I hope that I could show you that senescent cells are indeed quite common contributor to aging and pathologies associated with aging. However, the senescence program is very heterogeneous, and there might be a lot of compensatory mechanisms that would make senescent cells more or less resistant to the common senolytic interventions that we have. And I didn't mention this, but an analog of ABT263 is currently in phase 2 clinical trials for macular degeneration. This is a drug that Unity Biotechnology is using, or an analog of ABT263. However, as I mentioned, not all the cells might respond the same to this compound. So it depends really much on the tissue and on the disease that we're looking at. And I show you that indeed combining perhaps different type of approaches, for example, inhibiting BCL2 proteins and MCL1 anti-apoptotic protein could also synergize to eliminate certain type of senescent cells, in this case, senescent melanocytes. And this is a picture of our group, and of course, I mentioned, I think, most of the people that are here. Important to note, we also have a postdoc position. Many labs have advertised this, so if you fail with all the other good labs that have been there, please come to me. First of all, we are poor, and second, we are very cheap. So we don't do retreat in Portugal, I'm sorry. But what we do is that we actually spend a couple of days in North Holland, in islands in North Holland, and we actually walk in the mud. Because it's free, so we can do it. So we can do for more days if you want, as much as you want. And the last thing I would like to advertise, talking about North Holland, that we are organizing at the end of the month the meeting of the International Cell Senescence Association in Groningen, and of course you are all welcome to come. And thank you very much for your attention.
Thank you so much. Really fantastic talk. There's a question from Will.
Thank you very much. I was wondering about the senescent phenotype in a malignant melanoma versus the normal pigmented cells. Do you see a different senescent phenotype there?
Yeah, so yes and no, in the sense that what we also tried, because this was another approach, and then it's another approach we are taking. So we took a melanoma cell and we treat it with chemotherapy, for instance, and we actually make the cells enter a senescent state. And what we see is that these cells are exactly similar in how they respond to the senolytic treatment. So they are resistant to ABT263, but they're sensitive to the combo. So even if they are already melanoma, they still maintain some sort of imprinting for having this type of resistance to apoptosis. There we didn't compare directly the two. Yeah, so yeah.
Great talk. So, question, maybe I missed it. So did you check if the aged mice senescent melanocytes, do they exist in the aged mice? And did you try for the treatment there, and can you remove it?
Yeah, so one of the problems that you have is that mice, so we think that this is quite specific. We didn't do the same, the real experiment. I mean, it's a little bit difficult. Also because mice has very sporadic melanocytes. It's not like humans. It's very different. So that's why we use this trick where we have these transgenic mice where we induce the hyperpigmented lesions. Because otherwise it's very difficult to go cell by cell. And there are not so many senescent melanocytes that happen. In humans, it's completely different in terms of... It's not like the same though, you know, and especially high doses of UV radiation cause completely different phenotype than chronological aging in mice. So it's very tricky to make, unfortunately, that's a limitation of the skin in terms of translating between mice and humans.
Yeah, I find it very interesting that senescent melanocytes are more resistant to apoptosis. So I wonder, are melanocytes, like even like healthy young melanocytes, more resistant to apoptosis, or is this a property that appears with senescence?
We did some tests on this, but honestly, we didn't see that they are more resistant. So if you titrate, you know, classical pro-apoptotic drugs and you compare with how melanocytes and fibroblasts react to them, the curves are very similar. So I don't think that they are intrinsically resistant. It's really a program that starts with this, how they enter senescence.
So do you think, sorry, just very quick, do you think is there any like evolutionary reason why? senescent
Well-
melanocyte would persist in these molds?
That's for sure, that's for sure. This is a question that I think in the field we haven't explored enough. But for example, why do we have nevi, right, that actually are there for decades? These are indeed nests of senescent melanocytes, right? There is no other tissue that would have this nest of senescent cells all the time for so long. Something will take care of them. But nevi are very special. So evolution, there is something which, we don't understand. But for us, melanocytes, of course, are very important for the UV stress, so to protect from UV stress. So perhaps that might be something there that we need to explore better.
All right. Thank you so much, Marco. That was really fantastic. Amazing talk.